Okay, so this is figure 7, and it illustrates how researchers create a universal donor cell by engineering them to evade all major arms of the immune system. So first, they delete the B2M, so the cells no longer display the HLA class 1 molecules, preventing CD8 plus cytotoxic T cells form, recognizing and killing them. Because cells lacking HLA-1 would normally trigger NK cells, the engineers add back a protective signal, which is HLA-E right here, which binds inhibitory NK receptors and prevents an attack. To stop recognition of CD4, they knock out the C2TA right here, eliminating the HLA class 2 expression. The cells are also protected from macrophages by overexpressing CD47 here. They basically give out a protective signal, like a don't eat me signal that inhibits phagocytosis. And then as shown in figure 7b, these combined modifications make the cells highly resistant to immune rejection in both lab assays at the top and mouse models at the bottom. The engineered cells survive longer and remain functional even when donor and recipient.
Okay, so this engineering strategy offers several advantages for creating universal off-the-shelf cell therapies. By removing HLA class 1 through BM2 knockout, the cells avoid destruction by CD8 cytotoxic T cells. And adding HLE prevents NK cells from launching a missing self-attack despite the absence of normal HLA-1. Knocking out C2TA further reduces immunogenicity by eliminating HLA class 2 and blocking CD4 T cell activation. The inclusion of CD47 provides a don't eat me signal that protects the cells from macrophage mediated clearance. Together, these modifications improve cell survival in both animal studies and mixed donor cultures. Because they eliminate the need for HLA matching and patient specific manufacturing, these universal cells could be widely applied across many therapeutic platforms, including CAR-T therapies, stem cell derived products, and iPSC based tissues or organs.
So, of course, this approach isn't perfect and it's going to come with several important limitations. So, when we make these cells harder for the immune system to detect, we're also going to introduce new safety concerns. For example, removing HLA molecules and dialing down non-killer cell activity could make it easier for harmful mutations, viral infections, or abnormal cell growth to occur under the radar. Then there's also the issue of CD47. So this is typically used to protect the cells from macrophages, but tumors actually use the same exact trick. So if we push CD47 expression too high, we could potentially interfere with normal cell cleanup processes or even trigger unwanted inflammation. And another challenge is going to be the engineering itself. So these cells require multiple knockouts and knock-ins, which means a lot of genome editing. This raises the chance of off-target CRISPR effects and makes manufacturing even more complicated. And even after all these edits, the cells still aren't completely invisible. Minor antigens, cellular stress signals, or existing immune memory can still cause rejection. Non-killer cell avoidance is especially tricky because HLA-E has to be expressed at just the right level to prevent an attack without suppressing the immune system too much. And finally, there's the regulatory side. So cells with extensive editing face more scrutiny, and removing HLA class 1 may also weaken antiviral responses, making it harder for the body to detect infections inside these engineered cells. So while this strategy has a lot of potential, it also is going to require careful balancing to keep it safe, effective, and clinically feasible.